Hey guys, welcome back to One Stop Biology. So guys, till now we have studied what exactly? So we are right now at the chapter 13 of class 11th NCERT. We are right now at photosynthesis in higher plants, and till now we have understood two major processes. So we have understood the basics of what pigments are involved in photosynthesis, what is the reaction which is exactly happens in photosynthesis, where does it happen exactly. So we also studied the structure of chloroplast. And then in the last two videos, we have studied non-cyclic and cyclic photophosphorylation, right? So we studied how the energy molecules, the ATP or NADPH molecules are synthesized in the photosystems, the photosystem 2 and the photosystem 1. Some of the processes are non-cyclic, while some of the processes are cyclic. Now, today we are going to study about the chemiosmotic hypothesis. Now, what is exactly this hypothesis is? So basically, this has put forward to explain the exact mechanism of how actually ATP is synthesized in the chloroplast. So basically, till now we have just seen that the electrons are transported from photosystem 2 to the electron transport system. From there, it moves to photosystem 1, and there it converts ADP into ATP and NADP plus to NADPH, right? Now, today we are going to understand how exactly this conversion, how exactly this synthesis of ATP happens, whether it's a non-cyclic process or it's a cyclic process which happens only in photosystem 1, right? So, guys, like in respiration, even in photosynthesis, ATP is synthesis is linked to development of a proton gradient. So a proton gradient is developed across the membrane, right? Now here, what are the membranes which are involved in synthesis of ATP? It is the membrane of thylakoid. So I hope you would have gone through the videos, through the previous videos that I have made for this chapter where we have seen the exact structure of chloroplast, and we also studied the structure of thylakoids, right? So basically here the proton gradient is being developed across the membrane of thylakoid. Now here there is one difference though, that the proton accumulation is towards the inside of the membrane. So basically the proton accumulation is in the lumen, right? Now in respiration, this proton accumulates in the intermembrane space of the mitochondria, right? So when the electrons move through the electron transport system, in respiration, what happens? This respiration we are going to anyway, which we studied in the next chapter. But what happens? This proton accumulates in the intermembrane space, right? Now here the proton gradient is across the membrane and the proton is accumulating inside the membrane which is in the lumen basically, right? So basically what happens now, this in this process what is happening, activation of electron happens, and then the transport of electron determines the steps that causes the proton gradient to develop basically, right? So it's all about this movement of electron. Now since splitting of the water molecule takes place in the on the inner side of the membrane, right? The protons or hydrogen ions which are produced by splitting of water. So in last video we also saw the splitting of water wherein two molecules of water are splitting into what? Four hydrogen atoms which are protons plus molecule of oxygen plus four electrons, right? So basically this accumulation of proton, this accumulation of this hydrogen ions are basically happening in the inner side of the membrane. So basically when the water is splitting, the protons are getting accumulated within the lumen of thylakoid, right? After that, what happens? electron moves through the photosystems, right? First is it moves to the photosystem 2, and then from there it moves to the electron transport system, the electron transport chain, and then from there it moves to the photosystem 1. So when this electron moves through the photosystem, these protons are transported across the membrane now, right? So basically this happens because the primary acceptor of electron, now this primary acceptor of electron is located where? It is located towards the outside, the outer side of the membrane, right? And this transfers the electrons not to an electron carrier, but to an proton carrier, right? So this basically transfers, this primary acceptor transfers the electrons to a proton carrier, right? Now that is why the molecule removes a proton from the stroma while it transports an electron. Now, when this molecule passes on its electron to the electron carrier on the inner side of the membrane, the proton is released into the inner side or the lumen of the membrane. Now, after that, what happens? The NADP reductase enzyme is present in the stroma, right? So that is located again on the stroma side of the membrane. And along with electrons that come from the acceptor of basically electrons of PS1, these protons are necessary for the reduction of NADP plus as well. So this proton is required for reduction of NADP plus to NADPH plus H plus. So we saw this reaction as well. So this happens and in the presence of proton as well, right? So basically the protons are necessary here, and then these protons are also removed from the stroma. So basically what we are saying that within the chloroplast, protons in the stroma decrease in number, while in the lumen of the thylakoid, there is an accumulation of proton, right? So this creates a proton gradient across the thylakoid membrane, as well as there is a measurable decrease in pH, where in the lumen. Right. So this gradient is very important, guys. This gradient is important because it is the breakdown of this gradient that leads to synthesis of ATP. Always remember that because of this gradient, because of this breakdown of this gradient, the synthesis of ATP happens, right? So basically the gradient is broken down due to the movement of proton across the membrane to the stroma through a transmembrane channel, right? This happens through a transmembrane channel of CF0. It is known as CF0 of the ATP synthase. So basically ATP synthase is an enzyme which has two parts, right? One part is CF0, which is embedded in the thylakoid membrane, and it forms a transmembrane channel. Now this carries out facilitated diffusion of protons across the membrane. Now the other portion of this ATP synthase is CF1, and this protrudes on the outer surface of the thylakoid membrane, which is on the other side, on the other face, right? So basically, it basically faces the stroma. Now, the breakdown of the gradient provides enough energy to cause a conformational change in CF1 and part which is the particle of the ATP synthase enzyme. And this makes the enzyme synthesize several molecules of energy packed ATP, right? So we will see this complete reaction through this flow chart as well. Let's see this flow chart once. Right. Now just let's assume that this is the thylakoid membrane. Okay, this is the thylakoid membrane. Now, what is happening is light is falling. This is light which is falling on the PS2. Now this is basically P680, right? And this is it's falling here. And in PS2, what is happening? In PS2, the splitting of water molecule is happening, right? So basically we have oxygen plus proton, right? And then there are electrons as well, right? Now, because of the splitting of water, we have electron which is getting excited, and it is doing what? Basically, it is getting used by this plastoquinone. It is getting used by this plastoquinone, right? And it is also using proton, right? And it is getting transferred to where? It is getting transferred to the electron transport system, which has cytochromes. So here it has cytochrome with the name B6F, right? So from here, this electron is getting transferred to PS1. And here again, it is using plastocyanin. These are all molecules, right? Again, here light is falling. And this is our PS1, which is P700. So it is basically absorbing wavelength of 700 nanometer. Now here again, what is happening? We use various enzymes like FD, we use FNR, right? And NADP plus molecule is getting converted into NADPH, right? So here we have NADPH molecule being formed, which is an energy storing molecule. And here again, we are using protons. Now, what happens? There are several protons released now. So basically, from here, proton is getting released. From here, proton is getting released, and both of this. So there are a lot of protons here inside the membrane of the thylakoid, right? And this proton is going to the ATP synthase enzyme, which has two components, CF0 and CF1. So these are the two components. One is this, which is CF0, and then the other one is this, which is CF1. So this is basically ATP synthase. And what it is doing is, again, it is using proton, and the ADP molecule is getting converted to ATP, which is again the energy storing molecule. Now, what is happening? Outside you have the stroma, right? And inside you have the lumen. Now, lumen has high concentration of protons, right? And basically, if you talk about the electrochemical potential as well, it is high towards this side and it is low towards the other side. And because of this proton gradient, synthesis of ATP is happening here. So because of this proton gradient, there is changes in the CF1, the other portion of this ATP synthase, right? Because of this, there is a conformational change in CF1 particle, which makes the enzyme synthesize several molecules of ATP, right? Now, guys, this chemiosmosis requires a membrane, right? So we can see a membrane. There is a proton pump. So there are proton pumps present. There is a proton gradient, which is higher here and lower here, right? And then it also requires an ATP synthase enzyme. So the energy is used to pump protons across the membrane, which creates a gradient or a high concentration of proton within the thylakoid lumen, within the lumen of the thylakoid. And then the ATP synthase has a channel that allows diffusion of this proton back across the membrane. So again, this releases enough energy to activate the ATP synthase enzyme, and that catalyzes the formation of ATP. Right? Now, along with the NADPH, which is produced here by the movement of electrons, ATP will be used immediately in the biosynthesis reaction, which is taking place in the stroma, and that is responsible for fixing carbon dioxide and synthesis of sugars. So, all that we are going to study in the next video. So, guys, I hope you understood this process of chemiosmosis properly. If yes, please do like the video and share with your friends. And if you have any doubt, please do not hesitating in asking your doubt. Please do mention your doubts in the comments of the video and give your feedbacks. And yes, guys, if you're new to the channel, please do subscribe to the channel and press the bell icon to get all the updated videos that I'll be posting in future. Thanks for watching this video, guys. See you in the next one. Take care. Bye bye.